very short introduction, and I will try, but it's hard because he is one of the most extraordinary scientists that I have met in my career. We met each other when we were young and happy at Harvard Medical School, and Jerry moved on to the Carnegie Institute in Washington, and I went out to Washington, St. Louis. He remembers me visiting his lab, being totally ignorant of recombinant DNA, and he advised me on some topics, because we were getting into the biochemistry and recombinant DNA. But Jerry has been interested in Drosophila from many, many different points of view, not the least of which was he led the effort to sequence the Drosophila genome, which today sounds pretty straightforward, but at the time, it was the first complex genome sequenced. And it was a tense time between the government consortium and the private industry with Craig Ventnor. And what Jerry did was show that Ventnor's methodology worked really well, and in fact was adopted soon later by the government sequencers. And the Drosophila genome has been pivotal in many areas of science, especially developmental neuroscience and developmental biology and neuroscience. Jerry spent time studying at Berkeley and at Stanford, where he did a postdoc with David Hogness, and in England with Brenner in England. And he's followed in the footsteps, but much more so, much more molecularly than Seymour Benzer, whose book, Time, Love, and Memory, I recommend to everyone, which showed that you could use Drosophila to study behavior when many molecular scientists thought it was a waste of time. But now it has become a centerpiece, not the centerpiece, for the Howard Hughes Research Institute at Genelia Farm, an institute that Jerry heads. He's currently vice president of the Howard Hughes Institute. And I am astounded at how well Genelia Farm has grown from a large, elegant, somewhat isolated institution to a very vibrant community of interactive scientists based on very unusual models of research groups. And I think Jerry's influence is evident in every one of those groups. So Jerry, come on and give us your talk on Drosophila. Is that short enough?
No, but it was very nice, but it wasn't short. So I'm going to try to tell you a story about using Drosophila to study neurobiology. The title here is somewhat unusual. Why would you have a title like this? And the reason is that I am a molecular geneticist. I'm not a neurobiologist. I got involved in Genelia as an experiment in sociology. I still, if you ask me what I spend my time on, I say I do sociology of how to get creative people to work together to do interesting things. We started, the Howard Hughes Board of Trustees approved building Genelia 3 years before we had decided what were the scientific problems we would study there. So the basic sociological model, the building was designed, the construction was halfway done before we had our first planning workshop to say, well, what areas of science, what problems should we tackle? So at that point, we picked neurobiology, very basic neurobiology at the level of identified cells, and imaging, making better microscopes as the 2 areas we picked. Neither of those areas I'd ever worked in, but I realized I'd better be a useful member of the society. So I thought, what did I know how to do as a molecular geneticist that would be relevant for neuroscience? So I began to think, okay, how do I work as a geneticist, and could we use that model to study the brain? And I said, well, in genetics, the gene is the unit, and maybe in the brain, the cell is the unit. The cells are complex, but genes get phosphorylated and otherwise modified and have differential splicing project. So maybe that was a reasonable thing. And in that model, you would say, well, what do you want to have? You want to have something that's equivalent to the genome sequence. That would be a wiring diagram. And then you'd want to be able to manipulate the cells in that wiring diagram the way you can manipulate genes. So you need to have a way of directing exogenous proteins and RNAs to individual cells. That's the tools you use to manipulate those cells. So we had to make transgenic animals that would express things only in particular cell types. Okay, so once you have those, you can use them to express proteins that will modify the function of those cells, and then that's like making a mutation, and you can analyze then the effects of those by doing behavioral assays. And then you can do the equivalent of biochemistry, electrophysiology, or functional imaging, or other methods to look at the function of cells. So this is a simple analogy I had in my head, and I said, well, I can work on this second part, because this is a genetic engineering project, making drivers that are cell type specific. Now, the reason to do this in Drosophila is illustrated in this slide. Drosophila brain is small. This is a scale picture of a whole Drosophila brain and one cortical column in a rat. So if you're going to do things like electron microscopy, which are very slow, you might as well tackle something. You know, and already I was getting old, so I wanted this to happen in my lifetime. That was another motivator for working on Drosophila. And so Drosophila only has 100,000 neurons. This is still a lot more than C. elegans, which has 300, but Drosophila has a lot more complex behavior. So it seemed like a nice balance between complexity, behavior, interesting computational problems that the nervous system has to accomplish, and tractability. Okay. So where do we begin? I'm going to tell you two stories this afternoon. One is a very simple story about sensory perception, something where the nervous system is pretty much hardwired at birth, but there are interesting computations done. And then I'll spend most of my time talking about learning and memory, something where there's plasticity and the animal has to adapt to its environment. Okay, so this is a diagram of part of the Drosophila nervous system, the optic lobes that process visual information. And flies have those facets. They have a very repeating structure, which takes retinotopic information from the world, so it preserves where things are in space. But further on in the nervous system, there are cells, which are shown here in this cartoon, which lose their retinotopy. They give away all the spatial information. And so people have postulated that they must be taking information that's processed upstream and then computing some salient feature the fly cares about. Something's coming to hit me with a fly swatter, or there's a potential mate over there, or something like that, and conveying that to the brain, because it's no longer has the spatial information where in space it is. So we decided to study those cells. And so now we come into the genetic engineering. So for many years, I studied how gene expression is controlled. I had many graduate students at Berkeley in the 1980s who analyzed gene expression sequences. So I sort of figured I knew how to do this. And genes are controlled by pieces of DNA called enhancers. So there's this fragment of pink DNA here, which is an analog computing device for the abundance and distribution of transcription factors in cells, and then gives us output of whether a gene is transcribed or not. And so we started collecting these fragments and making transgenic animals and assaying in the brain where they would drive expression. And we made 7,000 of those lines. And they gave relatively sparse patterns of expression. In other words, they were expressed in 100 cells out of 100,000, so 0.1%. But they weren't precise enough to be single cell types. Then we found that a simple intersection, by asking that a cell only drive expression if it simultaneously expresses two of these enhancers, we could get to the desired level of specificity that we needed. And I'll show you an example of this. Here on this side are two of the original lines that have a single enhancer driving in a pattern that reach a couple hundred cells out of the 100,000. And if we ask for the intersection of those, you get this. This is this few cells you get. It's two cells, one on either side of the brain. The brain's bilaterally symmetrical. So we can make these patterns that are this specific. It's all engineering. I'm not going to, it's all published. If you want to know how it is, you can ask me later. I'll tell you the references. But this was 10 years of my life, or the life of my small lab at Genelia. But we got to the point where it works, and we have a large number of the A and B halves that we can combine, and we're just now making these for the various cells we're interested in.
But you said the intersection
Mm-hmm.
between things, different halves of the brain?
No, sorry, I was confusing. We produce one half of a transcription factor driven by one of those enhancers. It's not active, so it's only half the protein. And we produce the other half with the other one. So if a cell expresses one or the other, nothing happens. Only if a cell expresses both do you get an active transcription factor. So it's a molecular intersection. Yeah, sorry. Yeah, you shouldn't interrupt because I'm not sure of, the background of everyone in this audience is a new kind of audience for me, and I'm trying to explain things without using too much jargon, but, I don't want to lose anybody.
But the intersection transcription factor may in fact not be found in nature.
That's right. I'm not care about nature. I'm making a tool. I'm an engineer. That's irrelevant. What I care about is what I can assay directly that it's only expressed in those cells. And all I care about is if I take 1,000 fruit flies of the same genotype, and I look in their brains, they'll all be the same and it'll be reproducible.
Is this cell type unique in that there's only one instance of that type on each hemisphere? Or is it?
That depends on your definition of cell type, which is a problem best discussed over lots of alcohol.
I meant when you do crosses, do you get sometimes multiple instances in a cross?
Well, there are some cell types that have more than one in animals. So in the eye, which is a repeat structure of 800 columns, you get a cell type that will be 800 times on each eye. Right. Okay. So you can get those. All right. So then we made these cell type specific lines for the various cell types of these lobular columnar projection neurons. And these are just different fruit fly brains where you see the cell types in green, and you just have that cell type. So now we have the tool we need, and we do lots of light level anatomy. So this is a part of a fly brain where the gray is just a reference stain, so you can see the whole brain. And the green is this one cell type, has its dendrites in the lobula here, and all its axons coalesce in one place. It's throwing away the spatial information. And if we label with something, only label synapses, we know that's the part of the neuron where the synapses are. And we can label individual cells in different colors using genetic tricks so we can follow individual cells. So we can characterize what these cells look like. And there's a whole collection of these cell types, all sampling information from the full range of space, but then coalescing each cell type, its output, to a small point in space, throwing away the spatial information. Okay, and we can do what they are, and this shows it throws away the information, and they have different layers. So each of these cells is getting different information from the outside world. So in the optic lobe where their dendrites are, where they get their synaptic input, they're in different layers. So something's going on here. There's a computation going on from the inputs to these that we think is going to give some salient feature, and I'll show you some information for that. The fact that they go to different layers here just says they're getting different input. Okay, and we can characterize that. Okay, so now we have these, and we can now, what happens if we now go on to the next step of manipulating these cells and looking for changes in behavior? So there were 2 postdocs involved in doing this, one in my lab, Ming, and one of my colleagues, Gwyneth Codd and Ryan, and they did a very simplest experiment you can do. Take these neurons that has one of these cell types and just activate it. Okay, we do this with optogenetics. We can make ion channels that are light sensitive and express them in these cells and see what happens when you activate these cells, and we can screen these different types. And I'll just show you an example of this cell type here. So here's a bunch of flies, and they're walking around, and the light comes on, and they all back up. So this, you would say, okay, this may be telling the fly there's something to be avoided. All right, and then there are other ones. This is the backward one, and then the other ones where they stop and they reach, and there's ones where they just sort of jump around like popcorn. Okay, so, and the reaching looks like this at this high speed, 6,000 frames a second video. They get up and they reach out. And this is a motion that flies do when they come across a gap in their terrain they have to cross, or during mating, and they reach out and male flies touch the female flies. So, and then this one that was jumping really shows the same thing as you get when the fly senses a looming stimulus, like in its escape response, and all the details in high-speed video are the same. So activating these cells are triggering responses, behavioral responses. And these responses are very stereotyped, but we're providing a very unnatural stimulus here. Fly normally wouldn't ever uncover the entire outside world being activated in one cell. So already, if you just vary the stimulus to the one that backs up like this, if you just activate the cells on this side, it will turn away from it. And the one that reaches, if you activate on one side, it will turn towards that side. So already this, I don't want to overinterpret what we learned from this. I think what we learned is that these cells are conveying different kinds of information from the outside world. And of course, an interesting thing is how the upstream cells in the outer part of the fly eye are putting inputs that these cells can calculate. This is the appropriate response. But the next thing that we want to look at is what are these cells actually responding to? Something a little more sort of natural. So there's a graduate student, my other colleagues, Michael Reiser's lab, and what she did was do calcium imaging the activity of these different cell types, 3 different cell types, while presenting different stimuli. So these here on your left are different stimuli. They'll play in a loop that are played to the fly. The top one is like a looming stimulus, what you'd get if you were being approached by a dragonfly that was going to eat you, or a fly swatter. The middle one is a similar kind of signal, but now it's a light signal on a dark background, which is, not natural for a fly. And then the bottom one is more or less a control for the top one. It shows increasing dark density, but now coming in as not with this edge like a normal looming stimulus. And you can see that the 2 cells that get backward walking and take off respond very strongly. The ones in the red and the blue respond very strongly to the looming stimulus, and they don't respond to an expanding area of light. They only respond to expanding area of dark, and they don't respond as well to just a darkening that doesn't have this expanding edge. So this makes sense. They're responding to that. And the black cell was just a cell we took that gave us no behaviors in our activation, but it's not inert because that cell responds extremely well to a small moving object in its vision. Okay? So these cells are tuned to different things, which means there's computations in the upstream neurons, of which have all been reconstructed at the EM level. I'm not talking about that. So one can begin to think about how do those computations take place. Okay, so that's a simple story as introduction to the kind of approach. It's the kind of hardwired processing for which simple invertebrate brains are well known for. Now I want to talk about a kind of problem which you don't normally think about in simple invertebrate brains. This is a problem of sparse coding, neural learning, memory, and it's a simple problem that all organisms that have to interact with their environment, and remember, they have to learn associations between stuff that's going on in the outside world and what's happening to them. So one way to look at this is there may be a bunch of parallel fibers in your brain. You have a lot of these, many more than the fruit fly. And they may have patterns of activity. And these patterns of activity on their own from birth, unlike the visual system where the things are hardwired to mean something, these have no meaning whatsoever. You have to learn the meaning from your experiences. Okay? What does it mean to you when neuron 2 is active? It has no meaning till you learn that. And how does this start to take place? Well, the general model for this, pretty much all learning, is you have input and output neurons from these. The input neurons are teaching signals. This neuron here. And when that neuron is active and this neuron is active, that coincidence of those two things causes a biochemical change at that synapse. And then that biochemical change is reflected by the output of that neuron changing. This is learning. This is the basic mechanism of all sort of learning at the very reduced logical level. So how does this happen? What are the rules by which this happens? Our thought was maybe in the simpler brain of a fruit fly, we could understand this. So the fruit fly has a structure called the mushroom body, which I'm going to tell you about, which has 2,000 of these parallel fibers. The simpler brain of the larval fruit fly, the maggot, only has 300, and the honeybee has 200,000. There's much more things it has to remember. But basically, we think we can study this using the tools we have and gain some insight. And for the rest of my talk, I'm going to be telling you about those studies. Okay. So in Drosophila, the major things the fruit flies learn is associating odors with whether they're good or bad for them. So in the nose of the fruit fly, there are olfactory receptors that are sensitive to different odors. These send their axons to the antennal lobe here, and the axons are the ones that smell the same odor, coalesce on the same target. And then something very interesting happens, which at first is counterintuitive. You throw away all this nice odor and you randomly connect those to these parallel fibers. So there's a lot of labs that have studied this, and there's a very nice theoretical studies by Larry Abbott and others that says this is exactly what you want to do for information coding. You want to randomize this information that gives you the best learning. And so this is a very conserved or ancient structure. And so those of you who are more familiar with the vertebrate brain, you could call this the nose, the olfactory bulb, and the piriform cortex. The diagram here would be the same. Okay, so so this is the mushroom body. So again, the idea is when a fly is presented with odor A, a small number of these 2,000 cells will be active. And people have measured that, and it's about 6%. And if you give them odor B, you might get a different set of those axons active. And so the fly can distinguish odor A and odor B. And this is what determines how many of these parallel fibers it needs by how complex things it needs to learn. So then, as in that cartoon that I showed you, you would have your teaching signals coming in and your output neurons. And we know from a lot of work that dopamine is the major teaching signal, dopaminergic neurons. And the idea from a model based on the work of many labs, I'm now sort of like summarizing like 100 scientific papers in one slide, that during this training, you would then, this is a model. I'm going to show you some data that's all consistent with this model, but in this one it's a model. You get this kind of synaptic plasticity at this coincidence of the odor signal and the dopamine signal coming in. And which of the Kenyan cells, these parallel fibers, are activated? That codes the identity of the odor, what the fly was smelling. And which dopamine neuron was activated tells the fly this is a good thing or a bad thing. Okay, the rest of the world says, when this odor is coming, am I eating nice food or am I getting an electric shock or whatever? And there are parallel sets of these units, and they encode different aspects of the memory trace, or the engram, that would have different stability. All right, so we wanted to test this model. So you can imagine now, if you just go back to the same kind of approach that we did before, the first thing we want to do is make the genetic tools that would drive in these different cell types. So we did that. It turns out why this system is great for doing this experimentally is because there are only 34 of these cells and there are only 21 cell types. And we can identify them all, and we can make genetic drivers for them. And so this is just a movie where you're seeing computationally aligning them all onto one brain. Okay, yeah, well, like I said, this is a hard question. For me, when this is not so hard because if something's on, there's 2 cells on one on either side, which is true for most of these because we have 21 cells, not 34 cells, they're one cell type. But if the cells look morphologically indistinguishable and the same intersection of these enhancers driving them, we're calling them a cell type. It's a very functional definition at this point. So what we found by a lot of light level neuroanatomy, which was published a little over a year ago, a couple of years ago, we found that, as expected, the Kenyan cells that run these long parallel fibers, but the output neurons, the dendrites, where they get their synapses, they tile along this, just like that compartment cartoon I showed you. And just like that cartoon as well, the outputs of the dopamine neurons also tile those parallel fibers, and those tiles coincide. That's why I drew the cartoon the way I did. That's the way it really is. And this is light level, and I'll show you later on. That's the way it is when you look at this at the level of electron microscopy reconstruction. So now we have this cartoon I've shown you before, but now we can say that the teaching signals are the dopaminergic neurons, of which, again, we found about 20 types. Here there are about 120 cells, divided in 20 types on each half of the brain. And then the output neurons are these mushroom body output neurons. So we have these tools to now begin to pursue. Yes?
Are you saying it's one dopamine neuron projecting to one output neuron?
In some cases that exists, but in some cases there can be 2 or 3 output neurons or 2 or 3 dopamine neurons. But they're small numbers. This is very, you'll see there are experiments we can do because of that you couldn't do in another brain. All right, this is a simple system, which those of us who were like came from the molecular biology or reductionists want to actually do experiments that we can control the variables. And so, all right, and we'll sacrifice a little bit of consciousness or whatever to be able to do these experiments. So the classic learning and memory experiment in Drosophila is you give an electric shock. This is what flies look like. You can tell, I didn't have to tell you when the electric shock came on. You can see the flies get a very strong stimulus. They don't like it. But this is not a very precisely directed stimulus. So the first thing we started doing about 2 years ago, 2.5 years ago, is doing that paradigm, but now taking one of these tools. This is a particular one of those output neurons. And what we did is went for each of the output neurons, if we individually inactivate that cell, and then say, can the fly still learn that when a to associate a particular odor and an electric shock and learn to avoid that odor. And when we did that, we found that we could get rid of 80% of the response by knocking out this one output neuron, 2 cells, one on either side of the animal. And consistent with the model, I wouldn't keep showing you this model if I was going to tell you it wasn't right, so you know, that this is a compartment, that if we inactivate the dopamine neuron to that compartment, and this is a case where there's one output neuron and one dopamine neuron, that we also block learning. And if we activate this dopamine neuron while giving the odor, we can teach the fly this adversive association. So it's both necessary and sufficient. We can write the odor just by activating that one neuron instead of the electric shock. So this is already much better than making the flies jump around like popcorn. Okay. Now we can get better now. And these are like, they're going to tell you some recent unpublished work. We can use optogenetic reagents, which are much less invasive an experiment. In that experiment I showed you, we use a temperature-sensitive ion channel. You had to raise the temperature in the flies, which has other effects. Here we can use light at a wavelength the flies can't see, but the channel can see, and the channel opens. And we use the specificity of the genetic drivers to give us the spatial specificity. So this is one of these genetic lines. It only expresses in those 2 cells. So we can just take a whole fly and stick it in a room and turn on red lights, and it's running around freely, unperturbed, and see what happens. So this is the nature of these experiments, and this is what this arena looks like, and we can play odors. We run odors in, and we can make quadrants in different odors, as you can see in this smoke picture here. So we have an apparatus where we can run odors in, and we can shine light, and we can film the flies. So what does that, a typical experiment looks like? We will flood that whole chamber with one odor, odor A, while giving them the light, activating this one neuron. Do that for a minute. Then take another odor B, and then show them that odor without the activating the neuron. And then ask them to choose between odors A and B. Okay, this is a very simple experiment. Of course, we do it in the reciprocal order as a control, but that's about the simplest learning experiment you can do. And this is flies in such an arena, and I'm going to show you a movie that starts just about when the air is ending, and you'll see the different quadrants with either an A or B light up. So here's just air blowing, now A and B, and you're watching the flies. This is real time, and they were trained to learn that A was bad for them by activating this dopamine neuron. And you'll see with time, they're exploring their little universe. Because they don't know there's a safe zone where they're just running around. But when they get there, they tend to accumulate in, and you get this distribution where they end up mostly liking to hang out in the B quadrants and not in the A quadrants. Okay? So we can, I'll show you that movie again now, but this time where we have machine vision tracking of these flies, and you'll see some trajectories. And it's interesting to see what actually happens. The flies explore. They decide they don't like it. They go back. They just, you know, somehow have to, they have no knowledge, of course, that there is a safe zone and a not safe zone. They have to explore their world. But you can see by their tracks that they are going around. If they go someplace where it doesn't so great, they often turn around. You can see this guy here going up, going around. The green one coming down. Hits the other odor and decides it's not a good place to be. Okay, so this is what we measure. And for the data I'm going to show you, I'm just going to show you a plot that plots where they are with time and the various things. I'm not going to, there's a lot of information in the terms and directories. We haven't analyzed that information yet. We hope to do that. But this just shows that with time, they're ending up in the B quadrant when they're trained that with this aversive neuron that A was bad. And we can measure this by just simple, the distribution in the A and B quadrant over the total number of flies. We call that a performance index. So this 0.7 here, minus 0.7 means there's 85 to 15 distribution. Okay, and we can measure that with time, and there's a plot of average and then individual trials. Okay, so now we can begin to measure lots of simple kind of psychophysics learning stuff that people have done on people or other animals. So first is retention. So we can look at, well, if I do this, say, 5 minutes, these are all the diagrams of the experiment. I just ask them, no more training, just ask them 5 minutes later. These guys remember fine. This compartment's involved in short-term memory. They won't remember anything a day later. So a day for a fly is like a human year, if you believe in like dog year kind of thing. So flies live about as many days as humans live years. So a day later, they don't remember this thing, but they remember, you know, it has about a half-life of a couple of hours. Then we can ask, what about extinction? Extinction is a phenomenon where you present someone after training with the two stimulus odors, but now with no reinforcing, no activation of dopamine anywhere. And these flies, they're not too bothered. There's a little reduction in the memory, but it's not pronounced. You can activate the dopamine neuron here without giving them any odor, and that causes the memory to be largely erased. Okay? And you can train them the other way around. If you now train them, give them another psychotraining, but the opposite. You train them, you give them the activated neuron when they're smelling B, they quickly forget the original thing they learned, and they're completely flexible and learn the new thing. Okay? So we can assay these things. There are other things we can look at. Memory capacity. Can they learn A is different from B and C is different from D or not? And can they distinguish similar odors? Like if you train them A versus B and they're very different, how does that learning work with versus A prime, which are very similar odors that overlap, that do overlapping sets of Kenyan cells. So these are the kind of assays we can do, and we can do them very rapidly. Not surprisingly, the specificity and the forgetting and the capacity are inversely related, and that's just follows from the sparse coding. If you have sparse coding and you learn odor A, and now you're learning odor C here, but you're simultaneously forgetting odor A, you can have a low capacity. You can remember one thing at a time, but you can remember that with great specificity. If you want to remember two things at a time, you're using up more of these sparse coding, and there's going to be some reduction in the specificity. Okay, this is what you'd expect, and this is what we find experimentally. Okay, so I've just shown you for this one neuron that it can drive fast learning. The retention time is only moderate. It can forget very rapidly if you give it dopamine alone without the odor. Has only a modest extinction rate. It can do this reversal learning of rapidly learning something new and forgetting the old thing. It has great odor specificity, very modest capacity, about 1, basically. Yes? Okay, so it's short, so you can assay the memory, like what do they remember 1 day later or 4 days later? So 4 days later is considered long-term memory in Drosophila. It's like, it's like 10% of their lifespan. Okay, so we have memories. This neuron can cause long-term memory, and I'm about to show you that other neurons and other compartments in the mushroom body do allow you to look at long-term memory, do encode that. So what I'm going to tell you is that the mushroom body has these different compartments, and they each do different kinds of memory separately, in parallel, initially.
All the dopamine neurons have the same aversive training associated with them?
No, some tell you to like the odor. So the valence and the stability of the memory are all specific for, I'm going to show you for different compartments. We're just getting there, and okay, so now we've done the same set of experiments with these different dopamine neurons that are all diagrammed up here going different here. And I'm not going to go through this in great detail, but you can see all these different criteria, they're different for these different compartments. So those learning rules are compartment specific. Okay, even though the odor signal is running through these long parallel fibers, it's the same going down the length of that axon as far as we can tell. So this is sort of, we weren't expecting this. This sort of makes it more interesting. The lesson I've learned is you look at something you think is simple, and things just turn out always to be more complicated. But it's then, there is pretty much not much else to do than to deal with it. So, but this is just doing those same experiments over, only instead of this dopamine neuron being activated, we activate these different neurons. And then there's different learning things. So there's single case of learning. So you might smell something, and at that time, it may be uncomfortable for you, but you don't know if that's a random thing. Is this real association just by chance? But if every time you smell that, a few hours later you smell it, you get the same thing, you might say it makes some sort of hand-waving sense for you to really remember that long term, because it's a consistent thing. So there are memories you can only form by these repeated trainings. And this is an example. You can take this neuron, and if you train it just once, you can get a small memory. But if you do repeated trainings, 10x, you get a very robust memory that lasts 4 days. This memory only lasts hours. And this memory can be erased by dopamine alone. This memory can't be erased by dopamine alone. So it's not just the neurons, it's the training regime. Okay, this is all going to reflect biochemistry into the cells, which I like because it's the area I actually know how to do experiments in, unlike neurobiology. But this is an interesting phenomenon. Okay. All right, so now let's get into a little bit into mechanism. What's actually going on in these cells? So here we had a very nice collaboration with Glenn Turner's lab, who was then at Cold Spring Harbor and has since moved his lab to Genelia, and his very talented postdoc, Toshi Hage, who's an expert electrophysiologist. So what they did is put a patch electrode to measure the response, the actual electrical response in this output neuron. And then in a live animal, they just shine, it's got a hole in it, so they get the electrode in. They shine light, and they're doing this exact same experiment. They're activating this neuron. And what did they find? What they find is that before the stimulation, that neuron responds robustly to either of these two odors. Okay, these are electrical recordings, okay, real electrophysiology. And after the training, now we're just training for a short period of time, over, here, 1 second, this is a 1 second interval. I was showing you 1 minute intervals in the live animals. When you pair the activation of the dopamine neuron with this odor, you greatly suppress the response of that neuron to that odor, but you don't do anything to the response to the other odor. So this is what you would expect. This is a specific learning. Okay, so that's the beginning of the first critical experiment. Now we're going to go in a little bit deeper. I want to talk a little bit about specificity again, and just say that at the physiological recording level, you get the results you would expect. If you have two odors like the two that we generally use that are very different, you get what I showed you. But if you have two odors that are similar enough that they actually overlap and activate some of the same Kenyan cells, which you can measure by imaging, then you get generalization. You get that behaviorally. The learning between the two isn't so great, and you get partial suppression of the neuronal spiking with the similar odor. So that makes sense. And what about the specificity of dopamine release? So in mammalian brains, sometimes dopamine diffuses great distances. Here, that doesn't appear to be the case. This is the experiment I showed you. If you now put dopamine in other compartments, not that compartment, you get no effect. And if you look in a different compartment, putting dopamine in that compartment, you also get no effect. So the specificity here, there's some recent experiments from Vanessa Ruder's lab using imaging, calcium imaging, which also point to this. There's specificity. These are local dopamine signals. Their effects seem to be limited to the compartment they go to. All right, now we're going to go on to timing. This is called associative learning for a reason. That is, you have to associate, like Pavlov's dogs, the two events. They have to be paired in time. So you have this learning here, which I showed you before, where you present the dopamine signal with the odor, you get that robust response. If you present the dopamine signal, then the odor, the fly doesn't learn to associate them, and you don't get any response. This is how the system should work. That's the way it works. That's great. Okay. Now, behaviorally, we've looked at this in much more detail. So here now, instead of 1 minute training, we're just giving 10 seconds training. And the cartoon shows up there, if the stimulus of the odor and the activation of the dopamine neuron overlap, you get robust learning. If it just immediately follows, you get robust learning. You start leaving it another 10 or 20 seconds further away, the learning gets robust. The fly doesn't learn that association so well. If it's right before, just like in the physiology experiment, it doesn't learn anything. But interestingly, if it's further earlier before, you learn something, but you learn something, the opposite valence. The fly is learning, oh, I get that odor immediately after this nasty dopamine signal comes in. This is like a good thing. The odor's a good thing now, and the fly learns to like the odor. It's called relief from punishment learning, okay, in human psychophysics. So flies can do this, and it's very interesting. This was known with the electric shock, but it's very interesting that you're getting this when we're just dealing with these single cells in this cell. So that's interesting. So we're finding a lot of interesting phenomena we'd like to understand here. So what are we going to do now? We'll look at some other data that's in that paper. I'll just summarize. The different learning rules are also reflected in the physiology. So this was a compartment that required slower learning, more repetitive training, and you get no learning with this 1 second paradigm of training. But when you do it for a minute, you go 2 minutes, you get robust learning. So it all fits. It's all consistent. Remember to point that. And in another compartment, which is long-term memory that's appetitive, we discovered, and again, this is published work, that there's a loop here that the output neuron actually sends its axons and stimulates the dendrites of the dopamine neuron and sends up this feedback recurrent loop. And by manipulation experiments, we know this has to be, all these components have to be working for several hours for the memory to form. After that, you only need the output neuron to read out the memory. You don't need the dopamine neuron anymore. Okay? It's very interesting. The system is getting more complex because all these neurons are talking to each other outside. So we have all these loops and other things going on. So again, I'm not going to, that all data is published. Okay, so now we're faced with this situation where it's not all the actions in the individual compartments. We know that this neuron I told you the most about sends its axons back into compartments of the mushroom body. And I've shown you loops, and I've shown you that some of the neurons leaving the mushroom body converge outside the mushroom body. So we really wanted to know at that point, what is this neuron that's so important for aversive learning? Who does it talk to? This becomes a problem we can't solve at the light microscope level. It has to be done by electron microscopy. So in the last part of my talk, I'm really going to talk about the field of connectomics, which is a controversial field in neurobiology, the way sequencing genomes were when it first started. People said, what are you going to learn from sequencing the genome? A bunch of A, G, C's, and T's. You'll never figure out what any of it means. So people say that about connectomics. And I think I'm going to make what I think is a very compelling case that that anti-connectomics argument is totally flawed, and we'll never understand the brain without having this kind of information. It convinces me anyway, so I'll see if I can convince you at this point. So there's a very active area of research at Genelia, which I am only a cheerleader for. So the work on the connectomics I'm going to tell you about is basically I'm standing in the sidelines waving a flag, but nothing more than that. So we are interested in the methodology, and we have some very good experimental physicists who are modifying electron microscopes to get better kinds of data. And these are two of them here, and we're doing something called focused ion beam milling scanning electron microscopy imaging. If you image the face of the tissue, and then you mill off a little more and image it again. And there are other techniques invented by Winfried Denk. We'll use a knife, a diamond knife to do that, but then you cut off bigger sections. Here we're using gallium ion beam. This is a microscope built for metallurgists that's applied to biology. That had to be modified because metallurgists do 2-hour experiments, and we needed to do 2-month experiments. So the whole system had to be put all these interferometry corrections and other things to keep it going. But basically, in the standard electron microscopy, you're mostly familiar with, you cut sections with a diamond knife, you make very anisotropic voxels. The voxels are very uneven. They're very good in X and Y, but they're 4 by 4, but now by 40 nanometers in shape. And so what happens is on the imaging plane you're looking at, up top it looks great, but if you computationally look at it from the side, you lose a lot of resolution. The advantage of this FibSim is you can make isotropic voxels. And so it looks the same from any dimension, which is going to be, we think, very important in computational analysis of this data, which is the rate limiting step in going from the images to making a wiring diagram. Okay, so there's a group at Genelia working on this, and we asked them if they would please do a piece of the mushroom body. So what's shown in this little cartoon up here is a lobe of the mushroom body, and this is actually going through the sections, and you can see this is the mushroom body there, and it's breaking up into the two alpha and alpha prime lobes. But anyway, this is taking 2 months of constant operation of the microscope. When it's done, computer can identify synapses in these pictures and can map you the density of synapses. And then with a lot of hard work of computer algorithms and people, you can actually trace all the neurons. And these are different output neurons. And you can see how they have sharp compartment borders, just like we saw in the light microscope. So what did we learn from this? So these are these parallel Kenyan cell fibers in this compartments of the mushroom body. And as part of these, there are some of these parallel fibers that don't get olfactory input. They sense temperature and light and other things. And then these other ones that sense that, and the number we estimated and the number you count in an electron microscope agree remarkably well. And if we look at one of these output neurons, we see that that output neuron connects to all the Kenyan cells. So these are the number of Kenyan cells, and the black numbers and the white numbers are the number of those Kenyan cells that make synapses onto that output neuron. And on average, they make 12, 14, 16. So they're heavily connected, and it follows a Gaussian distribution as if it's pretty much random, what you would expect, the range. So, okay, there's a number of cells connected and the average number of synapses from each of those cells. Okay. And the Kenyan cells also connect weakly to one another. We didn't know that before the electron microscopy. And those connections tend to happen where the Kenyan cells contact the output neuron. So you get these rosettes where this is the one output neuron, and these are all Kenyan cells making synapses onto it. And they seem to be also making synapses onto one another. This is a great mechanism for further synergistic sparsening of the signal and could have some real impact on the way the information is handling, but we have no data for that. That's an anatomical observation. Okay, so we see these Kenyan cells to mushroom body output neurons. Those were expected. They were in that model I showed you. There are two output neurons from this one compartment. This is the case where there are two. They're very similar. We call them the same cell type. EM reconstruction, they overlap and they fill the compartment. There's no difference in the connectivity. Okay. And we all know from the model that dopamine neurons should be making synapses on the Kenyan cells and then modifying the strength of the Kenyan cell to output neuron synapses. And again, we see that very high connectivity, but here, fewer synapses per cell. So these are the things that we expected to see. We saw everything that we expected to see. The surprise was, although there are 50 papers published a year on the mushroom body, it's been studied for 20 years, there are many theoretical models. All the theoretical models happily were based on these connections. None of them required any new connections. That's only half the story. And there's many and strong connections that were not expected and not needed for anybody's model. So this is my argument why you need the connectomics. So I'm going to show you what those are. No one expected that there would be Kenyan cell synapses back to the dopamine neurons. And in fact, these are stronger in numbers. We don't know anything about actual physiological strength, but in numbers, these are stronger than the dopamine neurons to the Kenyan cells, the ones that are needed for the learning rules. And the dopamine neurons also make direct contacts to the output neurons, as well as contacting the synapses. And while these are only about 500 synapses of a dopamine neuron into a compartment to that output neuron, where there are 13,000 synapses from the Kenyan cells. Remember that only about 6% of the Kenyan cells are active to any one odor. So probably the number of Kenyan cells that are active and the number of these synapses are active are of the same relative amount. So what these are doing, we have zero idea. We have no model. No theorist has ever said they should exist. And now we've challenged the theorists to try to think about what we should do when we're thinking about experiments we can test. But this, to me, is the biggest justification for connectomics, for any kind of science. When you think you understand something and you do an experiment that tells you you don't have half the basic irrefutable data in the wiring diagram, it tells you it was worth doing those experiments. As I tell the 10 people who spent a year of their life carefully tracing neurons in the computer, so it's easy for me to say. Anyway, all right, so now we have this data. We'd like to get more of it. As I mentioned, this is an area where we need more computation because I would say it's getting all the data you need to do a wiring diagram on a hard disk drive is certainly less than 10% and probably less than 5% of the effort right now. So this is a place where machine vision, we need a count on machine vision, even to do the fly connectome, let alone anything in humans. So, okay. Then there are these connections outside. They're far flung in the brain. So they're hard in this slow method of FibSim. It's great resolution, but it's slow. So we also have, no, sorry, I skipped a little bit here. But there are these are other connections. These feed-forward neurons here connect directly to the output neurons, also the Kenyan cells. These are these axons that go back in. There's the other axons that go back in. They do something similar. They talk to one another. And we have other neurons. I'm just saying all the neurons are in the volume we can account for. There are these inhibitory neurons that tune the whole sparse coding. If you get rid of them, coding isn't sparse enough. So we have these things. We understand all the neurons that are there. We can take these individual neurons now, because we have them labeled. We can sort them, and we can measure what genes they expressed. So this has all these subunits of the acetylcholine receptor. Lets us know that this receives acetylcholine as a neurotransmitter. It has the biosynthetic enzyme for GABA, which is good because our antibody stain says it was GABAergic. So everything is here. We have also other receptors and other things that we're looking at, like what is the receptor? When the dopamine neurons signal to regulate to this, are they actually using dopamine to signal? So we're beginning to look at that. And we've also looked at what the output neurons actually encode. So because this is where the sparseness of only having one of each cell, we can go back to animal after animal after animal, no recording from the same cell. We can't do that in any vertebrate system. And interestingly, flies are all different. The output neurons don't innately encode odor. Individual flies are different. This individual data is learned by plasticity. If you eliminate plasticity by knocking out enzymes that are required for that, all flies have the same thing. So the flies are very flexible, and we think the way this is working, untrained flies have a balance of positive and negative signals coming out of the various output neurons. And then what happens when you have this kind of plasticity, you upset this balance. So all this information has to be integrated outside the fly, and I'm going to skip some of this data that shows that you can activate output neurons and flies can be attracted or repulsed by that. And then to say we need to look at some of these things like where these axons outside converge. And so for that, this is an example of two output neurons, one that sends an aversive and one an appetitive signal. It looks at the light level. You can see the resolution here is quite good. It looks like they're going to have their synapses on a common downstream target, but we don't know what that is, and we'd like to know what that is. So this requires EM of this faster type with a transmission electron microscope, which we have a group at Genelia doing. And this is my last data slide, which is a movie which we're very happy with. This is a milestone achievement for us. This is a month old that we have all this data. This is all the complete EM data for an entire adult brain. It's 730 sections cut with TM, 21 million separate electron micrographs, 100 terabytes, and then it's a big computational task to stitch these together because the montage of images has to be stitched together in X and Y, and then the individual sections have to be stitched together, and they get stretched and distorted, so it's a complicated problem. But you can do that, and you can zoom in and get resolution down to seeing individual synapses. So now we have the substrate to test all those output neurons and where they go, and we just have to do that. And that's going to be highly manual and take a lot of effort from a lot of interested people, hopefully from all over the world. And so I'm going to stop there and just say the unpublished work on the mushroom body that I told you about was largely the work of Yoshi Aisho, a postdoc in my lab. A lot of the earlier work was done in collaboration with our friends at Columbia and in the Max Planck Institute in Germany, and lots of people at Janelia, and Glenn and Toshi, who moved to Janelia from Cold Spring Harbor. And again, what I've shown you is how we can combine all these different types of data in a simple system, a complicated system. And this is a bit that has to happen now because we're just generating data that we, frankly, cannot analyze. I don't think the tools are there, or the skills of the people that generate the data are different in the schools, or the people you have to map the data. So there's a plea to places like this that are interested in computation. We need help in the machine vision, how you go from electron micrographs to wiring diagrams, and then what does it all mean? And so I will end there. Thank you.